let's go for around 40 minutes. I think it will be fine. 45. All right. Hello, everybody. Hello, everybody. What can I say? I'm speechless today because we have a very interesting guest whose work I'm reading for many, many months now. And I would love to have him on for a millennia, if I can be honest. So I'm very joyful today for the wisdom that he will probably lay and explode our brains out of proportion. So he's none other than Dr. and Professor Pollack. Welcome, Dr. Pollack.
Well, thank you so much, and thank you for that wonderful introduction. I hope I can live up to what you promised. Um, Happy to be here.
I think you're already living up to that promise. The last person you need to reassure of that promise is me, myself. So I know you're humble though, and I appreciate you for that too, which is a
Well, thank
sign of men that I like to see when men are actually doing a lot of work and they are humble about it, which is something I have to learn. And I think young people have to learn to work and talk less. Right?
you.
Myself included.
Okay,
Um, s-
I hear you.
so, can you give to the couple of people that probably don't know you here? Can you give? Um, can I call it Jerry now? Is it
Yeah, yeah, that's fine.
to
That's
go
what
with Jerry? Yeah.
everybody does.
To the audience, this is note. Dr. Pollack was very gracious and he allowed me to call him Jerry. So that's it. So can you introduce yourself like for a few minutes and the work you're doing about water, and then I'll start asking a few questions of mine.
We discovered another phase of water. So we all know that water has three phases: a solid, liquid, and vapor. But we discovered there's actually a fourth phase in which the molecules of water are ordered, aligned with one another. And this is not a new idea, but it's been a controversial idea. And before I go into the background of this, um, uh, I should say that this is not a you might say a remote laboratory observation that has nothing to do with the reality, but is of interest only to a few scientists. This is the water that fills your body, and uh, the fact that it fills your body means that it's of some significance, and I'll I'll get to that later because uh, I think it's of extreme significance. So we had originally been studying the molecular mechanism of muscle contraction. You know how the proteins actin and myosin interact with one another to produce force. Um, and there was a guy who dominated the field, Sir Andrew Huxley, and he was a towering figure. He won a Nobel Prize for earlier work. He was president of the Royal Society, and when he walked into the room, there was a hush. It was as though God walked in. In the presence of God, you you keep quiet. And it was his theory. And unfortunately, we found in our experiments that practically every experiment we did, the results didn't agree with his theory. And we made considerable progress, but no, many of the people, uh, without without focusing so much on evidence, but more on people and such. If you had to make a choice between Sir Andrew Huxley, member of the famous Huxley family, and this guy from Seattle, uh, the choice is easy. So I would say that we contributed quite a lot, but still the prevailing theory is the one that sits with Sir Andrew Huxley.
Mhm.
And he passed about 5 or 6 years ago. One day, I was invited to a conference. The conference was in Hungary, and the conference was to commemorate the work of a famous scientist whose name was Ernst. And Ernst, he had passed a few years before, and his interests were both in muscle contraction and in water. Now, water was something that you know, we all drink it and such, but it had no particular significance for me scientifically. And I heard, and so I presented our work on muscle contraction, and um, the Professor Ernst was also a doubter of the prevailing view. So I think my presentation harmonized um, with the interests of people at the conference. But what really impressed me was the presentation by a guy named Gilbert Ling, and Gilbert Ling was not your average scientist. He came from China, and in 1948, the Chinese government decided that they were going to send their most promising young scientists to this country to get a further education. And they picked a physicist, a chemist, and Ling was the biologist. The physicist went on to win a Nobel Prize, and that's Xian Yang. And the chemist, I'm told, also won a Nobel Prize. I haven't checked that. And Gilbert Ling should have won two Nobel Prizes for all the work that he did, but he was too controversial. And Ling was maintaining, both theoretically and experimentally, he had a good deal of evidence that inside the cell, the water was not like the liquid water that I'm drinking from from this cup. Uh, liquid water, the molecules are randomly oriented, and they're bouncing around furiously many times per second or millisecond or even femtosecond. They're very actively jittering around. He said, no, that's not the case. In the cell, the water molecules are ordered. He called it structured water. And he said, well, it's obvious how this can happen because everybody knows who's into chemistry that the water molecule can be represented as a dipole. So what's a dipole? It's like a bean with a plus at one end and minus at the other end. And you can imagine how these beans would stack, would line up with the negative of one being attracted to the positive of another. And that went against the common views in which Brownian motion or thermal motion, the kind of jittering that occurs everywhere, would disrupt any kind of ordering. But Gilbert said, no, the evidence is against that. The evidence is that the water inside the cell is structured. And along with Ling, there were a dozen other people who had evidence to support the views of Ling, and I was really impressed. And what they were telling me, or what became obvious, is that if they were correct, then essentially all of biology was wrong because one of the central tenets of biology is that stuff moves around by diffusion. So, for example, an enzyme and a substrate, they're not necessarily next to one another, but they diffuse and bounce around, and by chance they come into contact. Well, with this kind of arrangement, that's not possible because it's like a crystal. Everything is oriented, and there's no possibility for things to get around. I was really impressed by that because this is big. So I went home and I didn't trust my own reactions or emotions, and I decided to give one of Ling's books to some of my students and postdocs. And the reaction was uniform. They all came back to me and said, "This is amazing. If this guy is right, then it's like the most practically the most important concept in biology, because a lot of biology would need to be revised." that intrigued me, of course, because there's a lot to harvest in that. So I decided I've got to get myself into it. I've been in the field of muscle contraction for a few decades, and I made my contribution. And this is far more important than muscle contraction. So the first thing I did was to write a book. It was published 2001. It's called Cells, Gels, and the Engines of Life. And the book devoted to presenting the ideas of Gilbert Ling, at least the first half of the book. And the second half went on to say, well, okay, what if Gilbert Ling is right? How does that impact cell biology? And I presented the simplest way that could happen, and that is, if a cell is activated, it's got let's say it's a muscle cell, and its job is to contract, right? So what happens, and I was able to marshal evidence from the literature, is that both the proteins and the water undergo a change. From the water goes from this structured kind of water to ordinary liquid water, and the proteins undergo change too. And it's a combination of proteins and water that precipitate the action of the muscle cell. Or if it's a secretory cell, it prompts secretion. Or if it's a nerve cell, it prompts transmission of information, etc. It doesn't matter what kind of cell. The cell turns on, and the water undergoes a transition, and the proteins together. It's called a phase transition, and it's well known in chemistry. And I was suggesting that biology works the same way as chemistry. So, and the reason I wrote the book is that Gilbert Ling's writings are really challenging to digest. Gilbert, who passed a couple of years ago at age just shy of 100, uh, uh, he was a brilliant guy, but his writing left something to be desired, and that is clarity. He'd sit down at the word processor, or before that, the typewriter, and he'd bat out something, and he'd send it to the publisher and say, "Okay, here it is, publish it." And the word editing was not in his vocabulary. So, then it was a real challenge to read and understand. And for average guy like me, I really had to struggle. And even for chemists who are involved, deeply involved in the field, even for them it was a challenge. So I tried to present it in a way that was understandable and palatable to the average scientist, shall we say, or even non-scientist. And the book was received with mixed reviews, unlike my later book, The Fourth Phase of Water, which seems to be wildly popular. But that book, um, some reviewers said, "Oh, this is more of Gilbert Ling." And Gilbert Ling, we all know he's a crackpot, so pay no attention to it. And on the other side, a well-known cell biologist from Harvard University wrote, "This is a 304-page preface to the future of cell biology." And that one I like better. Anyway, um, with that, I decided, we have to start doing experiments, because my laboratory is basically an experimental laboratory, and we started to study water. And so from there on, we learned a lot about the structured water in the cell, and actually even outside the cell too. And we learned that it was not quite the way Gilbert Ling put it. His general concept of a different kind of water is absolutely correct, but we found some differences experimentally. So here I am blabbering on. Maybe I should wait until you ask me the next question, uh, or
Go
I can just go on,
go
please.
ahead. Go ahead, Jerry.
Well, okay. So what did we find? Um, we found Well, first of all, we our original thesis was that the water, as Gilbert Ling had suggested, was a crystalline kind of water where all of the entities that build the crystal are um, regular, oriented, and in order to form a pure crystal from something, if the something contained contaminants, you got to get rid of the contaminants, otherwise the crystal won't be pure. It's like formation of ice. You know, ice is a crystal, and if the water that was used to form the ice contained contaminants, the contaminants would be excluded, would be pushed out as the crystal forms. And so we were looking for an experimental setup where um, contaminants, particles, whatever, are excluded. And we found it very quickly. And maybe by good luck or something, we put a gel into a little piece of gel into a chamber. We added water, and inside in the water, we added some contaminants, particles, little spheres called microspheres. And so you've got a gel with a flat surface next to the water uh, containing suspended particles. We looked in the microscope, and voila, uh, there we found right next to the gel in the water, we found a region, a sizable region that had excluded the microspheres. And 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 as we looked, that region grew bigger and bigger until it stopped growing. And so this was because it was excluding the microspheres. We called it the exclusion zone, and that works really well because exclusion zone, easy to remember. And it works, except doesn't work in every country because some people call it Z instead of Z. And so,
Yeah.
but we called it easy water, and that stuck. But in fact, we didn't know at the time. We weren't sure whether that water, which we called easy water, really was different from ordinary water until we started doing experiments. And we started doing experiments. We found every 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 property that we investigated of that exclusion zone differed from the property of liquid water. And so, given all of those properties that differed, we began calling it the fourth phase of water because it appeared to be a different phase. It was not ice and it was not liquid, it was some, something in between. So, fourth phase of water. And I'll just mention, I guess, a few of the most important features because they're relevant in pretty much everything I want to talk about. So the first is that we found that the molecules there are ordered, um, uh, and they're not random like in liquid water. It has a high viscosity, uh, sort of like raw egg white, and um, and surprisingly, it had negative charge. Now, water is neutral, um, and and by putting a tiny electrode into that zone, parenthetically, a microelectrode invented by none other than the same Gilbert Ling, for which he should have received a Nobel Prize because others later using that technique got Nobel Prizes. So at any rate, we found that that region was typically it was negatively charged, and and the first thing you think of, well, wait a second, water is neutral. How can you get a region of negative charge? The only way that you can reasonably explain that is there's another region of positive charge somewhere else, and we found that that was the case, and it was the region beyond this exclusion zone. Um, and so you got the gel or polymer surface, and then you've got the exclusion zone, and then you got ordinary water beyond that, and that water had positive charge. So you got negative in the exclusion zone and positive in the zone beyond that. It's like a battery. And by the way, we demonstrated that you can get electrical current from that battery. You stick one electrode in the negative zone, one electrode in the positive zone, and you can light a light bulb, or at least an LED light emitting diode. So, so then the question arises, if that is what I told you is really true. First of all, Gilbert Ling's idea can't be right because water, the water molecule is a dipole, and he was suggesting that you just stack the dipoles, and you can stack those dipoles. They're neutral, right? They're neutral. You can stack them from here to Antarctica, and you'll never get negative charge. So therefore, if our measurement is correct, and we've repeated it many times, it can't be right. And we found a different structure. It's a sheet-like structure. The sheets are parallel to the gel surface or polymer surface, and they stack upon one another. And we found experimentally stacks up to hundreds of thousands of sheet-like layers. So, so then the question arises: well, if it's a battery, if you have charge that's separated, everyone knows that their cell phone, if you forget to charge it at night, you got nothing the next morning. You got to charge it, and it's the same with every battery. All batteries need to be charged or recharged. And so, where does charge come from? How do we get? You can't get something for nothing. You got a battery, and you got to charge it. And we found that it arises from light. And the experiment that demonstrated that, it came from a young undergraduate student who was working in the lab doing the kind of experiment that I mentioned to you, a simple experiment in a chamber, a gel and water and microspheres. And he noticed that there was a lamp sitting next to him. He shined the lamp on the chamber. And where the chamber, the region that was illuminated, the exclusion zone grew enormously. And he called me in to watch, and I couldn't believe we had been trying so hard to figure out where does the energy come from, and there it was. This naive student who didn't really, he was kind of playing around, and playing around sometimes yields amazing results. So we then started to do careful experiments trying to figure out which wavelength of light is responsible, because that lamp generates a whole series of wavelengths. And we looked from the short end to the long end. At the short wavelength end, ultraviolet light, nothing. Visible light, the full spectrum from violet all the way to red at the long wavelengths, nothing. Maybe the reds at long wavelengths, just a little bit. And then we moved on to infrared, which is longer, the longest wavelengths. And there it was just amazing. Just a tiny amount of infrared light could build the exclusion zone enormously. And we've been able to, with a weak LED, to increase the exclusion zone size by a factor of 10. So it's really, really powerful. So anyway, the energy for buildup comes from infrared. At least that's one source of energy. And I think it's the main source of energy. So we reached the point. We kind of knew and understood the basics that exclusion zones grow. They have charge, and you can measure the opposite charge beyond them. And the growth is powered by infrared lights. It's a little bit like the first step of photosynthesis, where step one out of, I think, 20 steps, which are not really so well known, light comes. And the first step, which is 100% efficient, is that the light splits the water into OH minus and H plus. And then there are many steps beyond that. But the first step is so similar to what we found that we're still wondering whether what we found is a kind of generic first step of photosynthesis, where of course nature does it in the best way, and so the way that the plant does it is probably would be more efficient and more effective than what we found, but we may have found a kind of generality that applies also in the first step of photosynthesis. So maybe I should stop here and wait for a few questions because I think I'm using up all the time.
Well, that's why you're here, Terry.
Yeah, yeah.
I don't know about the audience, about the people watching, but I myself, I'm mesmerized by the whole thing, the whole journey, the whole process. And so you referenced. I'll put, for anybody interested, I'll put down links about anything Dr. Pollack, Jerry said today, till now and after that. But you said that there is the energy might be from the light, or it is from light, from infrared.
Yeah.
So I want to pose two questions. One is, of course, are there any other sources that can produce or provide the energy for this beautiful exclusion zone water to, or to happen? And the other question is,
Well,
what's the other question?
maybe I should answer that question while you think of the other question.
All
Otherwise,
right.
I forget. Yeah, we found that indeed there are other possibilities. So one possibility is to apply electrical current. So we found that if you apply current to the water in the form of electrons, electrons in from one of the electrodes, around that electrode, easy water forms. And it kind of makes sense because the easy water typically, as I said, is negatively charged. And so if you start with ordinary liquid water and you add negative charge, uh, we found that it converts to easy water. So that's one possible way. We also found that electromagnetic fields of certain frequency, not all frequencies, but certain frequencies, can build easy water. And my guess is that there may be more sources of energy that can build easy water, but we're at a
Hmm.
preliminary stage. We've just been studying it for maybe a decade or a little more than a decade, and there are many more studies to be done. So there may be sources of energy that we haven't even thought of that can build it. But yeah, so it's not just infrared light that does it, but infrared light's all over the place. A lot of people don't realize, but when it's like if you have an electric oven and you look at the coils, they're glowing a bright orange and they generate heat, and you say, oh yeah, that radiant energy, that's infrared energy. But in fact, it goes way beyond that because infrared energy is contained everywhere. So if in your studio, if we turn off all the light so that you can't see anything, and your cell phone camera, despite its high sensitivity, um, can't can't get an image of anything. If you have a camera that has instead of a visible light sensor, if it has an infrared light sensor, I'd get a beautiful image of the microphone and the pictures and uh, hanging on the wall and the lamp, etc. Everything is generating infrared energy, and that's why it's very convenient for the military to use it at night to see what's around, and they use exactly that technology. But what it means is that the energy that is responsible for building the easy or fourth phase of water is there all the time. Uh, it's
Hmm.
generating, and therefore you always will have some easy water. And in terms of inside your body, so you have all of this surrounding all the objects that are generating infrared energy, but even inside your body, the metabolism generates heat. So the core of your body is generating heat, and heat is not exactly, but approximately the same as infrared energy. So the infrared energy is being in terms of your body, it's being generated not only outside but also inside. So you're getting a lot of infrared energy, and therefore that infrared energy is responsible for building easy water in your body, and your cells are filled with it. Your cells are not filled with liquid water; they're filled with easy water. And just on that point, what one final remark is, you can test it yourself. If you're a masochist, you take a knife and cut your, or a razor blade and cut yourself. Now, if your cells were indeed filled with liquid water, and the water you cut yourself, the water would come out like a breached water pipe. You know, if you've had that experience, a lot of water comes out. But but when you cut yourself, the water doesn't come out. Plenty of blood comes out, but not water, not the cell water. And the reason is that your cell is filled with easy water, which is highly viscous, and that water sticks to the solids inside your cell, so it doesn't come out. It stays inside. And you know that is one way of demonstrating, and there are others, that inside your cell you've got easy water. You don't have liquid water. It changes essentially what we understand about biology and how cells work. So please.
So, do you mean, Jerry, that we are 70? They say, 70% water. So, are we 70% easy water then? And, you know, is all of the water in our body structured?
No, it's not. It's not all structured. Um, so, for example, think about the blood in your body. Well, first of all, the 70% water. That's 70% by volume, right? And if you estimate the amount of water in our body by counting water molecules, if you line up all the molecules in the body or in the cell, doesn't matter, and you start counting one by one, um, it's been estimated that more than 99 out of 100 molecules are water molecules, and the reason for that is that water molecules are small, and in order to fill that 2/3 or 70% volume, you need a lot of water molecules. So um, so the number more than 99 percent, if you think about it, is pretty significant, because in modern biology, the viewpoint and the textbook viewpoint is that water doesn't do a whole lot. It's simply the background carrier of the more important molecules of life. But if your cells are filled with easy water, and easy water transitions to liquid water and then back again, water is absolutely central to everything that happens in the body. So I guess I would venture to say that there's really some question about um, textbook concepts about how things work, and I think, based on what we found, if what we found is correct or largely correct, then a lot will need to be revised. And it's not all water in the body that's easy water. Um, we think that in the cell, it's essentially all excuse me, all easy water. But think about the blood. If the blood were highly viscous, easy water, it'd have a hard time flowing into tiny capillaries. So the body does what's necessary to ensure that the blood is actually not so viscous, and that the blood is effectively an aqueous-based system, where it's pretty much water. In the red blood cells, it's another another question whether easy water or not easy water. The red blood cells have the hemoglobin has two different states that are distinctly different from one another, and it may be that one of those states is actually easy water in the red blood cell, but the plasma that suspends the red blood cells, it needs to flow easily, and the same thing in the lymphatic system. In order for the fluid to move, it 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 can't be the highly viscous easy water. So yeah, I don't think anybody has really calculated to figure out how much of the body's water is easy water and how much is liquid water. I'd venture to say that most of it is easy water. There's more to say, but mm, please, please press on with your questions. Um,
I'll try. I'll try. That's very interesting, Jerry. And is there anything else about easy water that's important for ecosystems? Like, is this something that can be used besides humans, of course, which is us, very important to actually know that and be healthy? Are there any other animals and or plants and or parts of the ecosystems that have easy water and they need to have so that they can function well as we do?
well, absolutely. I guess the question could be rephrased to say what organisms in
Mm.
biology don't have easy water,
Ah,
because, well, and when we do laboratory studies, for example, diseases that we have, often we use rabbits or mice or whatever. The assumption is that they all function pretty much the same way. Uh, so, so whatever you find in a rat or a mouse or a rabbit, the presumption is it works pretty much the same way in the human. So, the humans are not unique. We're in the biological chain, and the same with plants. Uh, the systems are not exactly the same, but largely the same. And in fact, one of the expedients that many people use, people who are ill in some way, some um, of the health promoting professionals will say, "Why don't you do the so-called quote juicing?" So what's juicing? Well, you go to your backyard and you take the freshly grown plants, take the leaves, and you crush them and squeeze out the water and drink the water. And and usually what I hear from the practitioners is they come back in a few months and whatever was ailing them has is not ailing them as much or even at all. So what's going on there? Well, the freshly grown, healthy plant leaves, the cells are filled with easy water, just like we're filled with easy water. So when you squeeze out the water from inside the cells, you're basically squeezing out easy water, and then you drink the easy water, and it resupplies any of the easy water that's missing in your cells. Your cells must have easy water, because if they don't have easy water, they're not going to function as well. If function depends on the transition from easy water to liquid water and then back again. Obviously, if you don't have easy water to begin with, it's dysfunction. So your cells need to be filled with easy water, and one way to get the easy water is to drink it. For example, from plants. So yeah, obviously the plants have easy water the same way as the animals and have easy water, and we have easy water, and easy water is just all over the place, because whenever you have a so-called hydrophilic surface, that is a water-loving surface. That is, if you have such a surface, if you drop water, it spreads out because the surface loves the water and wants to interact with it, as opposed to a Teflon surface, hydrophobic, where the water beads up.
Mm.
It's the hydrophilic surface. Not every hydrophilic surface, but most of them build easy water.
Mm.
So yeah, in nature, outside, whenever you have a hydrophilic surface and water, you're likely to have easy water. So it's not just in biology; it's outside of biology as well. It's all around us. It's been around forever. It's just that only recently have we recognized it.
Interesting, huh? What you said now is very interesting. Most people think, and me included, that sometimes we discover something, and in full reality, it shows to us just how naive we are still for so many things around us. And
Yeah, well, don't get me into that, because it's not. I believe it's not only water, but that there's so much in the textbooks and in all fields of science. If something has been in the textbook for a period of time, we assume it's correct. Um, but when you realize how these things came into being, came from human beings. It came from someone like you or like me, who comes forth with an idea. And a few colleagues think, well, that's a pretty interesting idea. It gets into the textbook, and once it's in the textbook, it's really hard to eradicate, even if it's dead wrong. Um, and if it's been in the textbook for generations, then it's really hard to challenge. And I would venture that uh, there's a lot in the textbooks we believe to be correct that's absolutely not correct. If you want, I could enumerate some, but it's all over the place. And modern science is not geared up to um, effecting challenges. If you have an idea about any aspect of science, um, and you want to pursue it, the only way, convenient way, you need money to pursue it, because you need people, and you have to pay the people to do the work. Otherwise, they can't eat. You need the people, you need the equipment, and whatever. And the only way to get that, unless you know some philanthropist, is to get money from the government, from like the National Science Foundation, or National Institutes of Health. And think about it. So let's say you, Jason, you have an idea and you want to pursue it. And you have an idea that the Earth is round, but everybody around you knows that it's flat. You can just look out the window. It looks pretty flat, right? You can't really judge its sphericality. And let's say it's common belief, but you have an idea that it's round. And where does your idea come from? Well, you've seen satellite photos. It looks like it's curved, and you've taken a trip around the world, and you've been able to return to the place you start. So if the Earth is flat, it's hard to imagine that you can return. If it's spherical, then you can return. So you have this, which we scientists and the Jingo scientists call preliminary evidence, right? And you decide, okay, I'm going to write an application to get money, one of the granting agencies, and you say, hey, I've got preliminary evidence, and you cite that evidence, and you send in an application to, let's say, the National Science Foundation. And the gatekeeper receives your application and said, "Oh, I don't know this guy, Jason, but this is pretty powerful stuff. If he's right, it's pretty important, and therefore, I better recruit pretty good reviewers to check this out and see whether he's a crackpot or does he have something real there." So the gatekeeper recruits the biggest experts in the field of the shape of the Earth, and those are the leaders in the flat Earth theory, right? Because that's the prevailing theory of the day. And and they look at your application. They read through it, maybe, and they don't. They're not very happy about your application, because if you're right, they're wrong, and if they're wrong, then their reputation is basically obliterated. So your chances of succeeding, it's like the French revolutionaries, and they go to Louis the XVI and say, "We have we have a few issues with you." They're not likely to be received and embraced with enthusiasm, and it's pretty much the same in the scientific area. So most scientists know that if they have a radical idea, and some do, because these are intelligent, thoughtful people, but they know that there's almost no chance of success. And if they want to feed their families, uh, they'd better toe the line. And so, as a consequence of that, most scientists are doing what you might say mainstream work, checking out what sometimes we say dotting the i's and crossing the t's, rather than doing radical research that has the capacity to overturn a paradigm that it is prevailing, but yet has some problems and could well be wrong. And therefore, if I were to ask you um, to identify a single scientific revolution. I'm not talking about a technological revolution, like Zoom or like the iPhone or something, but a fundamental scientific revolution, something akin to the splitting of the atom or the structure of the genetic code, which are like 75 and 65 years old. One that's happened, let's say, in your lifetime. Um, I bet you would not be able to identify any, or more than a handful. And the reason is, I think the reason that I mentioned that the system is not oriented in such a way as to encourage that sort of thinking. It was 100 years ago, um, when there were no government organizations
Hmm.
to support science. The beginning of the 20th century, Einstein, Planck, all that happened in physics, for example, it's not happening now. And I think the reason is the reason that I described to you. So uh, my own viewpoint, which admittedly is not a mainstream viewpoint, is we need fundamental change in the way science is carried out. Um, there's so much to be learned and understood that we come to realize that the prevailing paradigms come from people like you and me. We ate the same food, we pee in the same toilet, et cetera, et cetera. People. It didn't come from the Oracle and Delphi or someone like that. Just ordinary people, and people are not infallible. So there we go. Anyway, a bit of philosophy.
Important philosophy, I would add. Interesting. And yeah, it's always sometimes the ego, the ego goes up, especially if you go into leading positions, and people get some recognition about something, and then they from what I see in my life, and they try to protect it. And
It's human nature. Yeah.
yeah, yeah, yeah,
And here we need to fight against human nature, because the very act of doing science means you're trying to find a revolution, something new, something that people don't know, or think they believe, but maybe they've got it wrong. That's what science is. But
yeah,
that's the way science is working today, unfortunately.
yeah, yeah. I think many people, or at least some people, got that memo the past few years. Anyways, Jerry, thank you for your time.
Sure, I'm very happy to.
It was my honor, honestly, and my pleasure having minds like yours here. And where, before we go, where can people actually find your work and your books and anything else you are
Well,
doing?
okay, so the books, I mentioned the names of the books. The most popular one is the most recent one. It's called The Fourth Phase of Water. And if you look on Amazon, you'll see the reviewers are really enthusiastic about it. It would be hard to find another scientific book with a higher rating than that. Um,
Mm,
and so that's what I would
excellent.
recommend. And anybody interested in the idea of water in biology, the earlier book. Cells, Gels, and the Engines of Life, also on Amazon. And if you're interested in and have the means to do something um, about the course of science, we have an organization of which I'm the executive director. It's called the Institute for Venture Science, and the idea is that people contribute, people of some means who want to see science really advance. We've designed the system to make it possible for radical ideas to take hold. And it's the Institute for Venture Science. If you want to look at the website, it's IV Science, IV like intravenous, but Institute for Venture, ivscience.org, not .com, .org. And there you'll see, and if you have the means and are convinced that this is a good way of advancing ideas that could really make a difference in our world, um, please contact me. Um, and of course, our own work in the laboratory, it's equally difficult to find funding. We had funding from a private funder who really likes our work. Unfortunately, he ran into some financial difficulty and couldn't continue. So there's so much going on in our lab that uh, stems stems from the work that I've briefly mentioned, but we need support to keep it going. So, if you're interested and have means, please email me. I'm easy email GHP at UW, that's University of Washington, .edu, GHP at UW.edu. I'd be happy to hear from you. So I think those are the major ways of uh, getting in contact and looking. If you want to look at our laboratory, it's called pollacklab.org. Real simple. You'll find our activities there. So I think that kind of sums it up. If um, that's okay.
Yes,
Yeah. Well,
yes,
thank you
yes.
for the
Excellent.
opportunity. I much appreciate it.
Yeah, thanks for finding the time, Jerry, and I respect you. And I'll put everything down below for anybody who's interested in supporting your work, because I think sometimes people in our age, maybe it's in our age, I don't know, but we seem to forget that people like yourself, people who are in on the forefront, you need support. Like sometimes people think it's freebie for everybody. Like these all these beautiful discoveries come out of thin air, like ether, and you guys drink ether for to sustain yourselves.
Yeah, wish that would. Yeah. No, we need funding. You know, a graduate student or a postdoctoral fellow for 1 year, it costs roughly $100,000, and to have a vibrant laboratory, you need a bunch of people. So it's not trivial, and it's really hard to get the funding when you're doing something that challenges the mainstream, because it's the mainstream that does the reviewing of the grant applications. And so it pretty much for this kind of work pretty much relies on private funding. And
Mm.
I was really tickled pink to have this donor for I think 6 or 7 years. He found us. We didn't, he said, I read your book, Four Phases of Water, and I really like it, and I want to help by offering funds. And it was just amazing for us. And
Okay.
but unfortunately, that financial obstacle made it difficult for him to continue. So we're lucky. Um,
We're hope is up. I think we can find another one and more. I think we can find more people, Jerry. Honestly,
well,
um, yeah,
if that's the case, it would be fantastic. We have
yeah.
so much more to contribute, and even practically out of. I talked about health just a little bit, but there are some technologies that come directly out of this understanding because we have an energy source, and we found we can get electricity from water and light, for example. We have a filtration device that if you collect easy water, it's free of contaminants. So if you put contaminated water
Mhm.
into a system, very simple, that we
Uh,
devised, and you
mm,
collect the easy water that forms, it's going to be free of the contaminants. So
mm,
you could, for example, we call it filterless filter because it actually is filtering out all the pharmaceuticals, the microplastics, and et cetera, et cetera. And you get drinkable water. And we even think that we can separate salt that way. So you could put ocean water into the system and get drinking water out of it, and it relies not on massive amounts of energy input, but only energy input from the sun. Um,
wow, can you imagine that? Huh?
well, there are lots of things on the agenda, but all of these need development, which requires money. And yes,
Well,
so there we are.
the Greek islands would be the Greek islanders would be very 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 interested in that. The last one you say,
Not only the
um,
Greek islanders, but everybody in the Middle East. You know, yeah,
true,
sure. Um,
true.
yeah, they've lots of salt water, ocean water around them, but not much water to drink for sure.
Yeah, yeah, yeah. Yeah, excellent, excellent. Jerry, please stay for 60 seconds. I'll after I stop the recording, and anybody else, please, if you like this beautiful chat that we had, and I think it's groundbreaking, and we should give it the meaning that it has and the gravitas, I would say. Please go down below, like, subscribe, and of course, and all the rest. But go and find the links and the email, and if you can support Jerry, Dr. Pollack, and his colleagues and the staff, please consider doing so because it's important work. Having said that, till the next one, guys. See you.